Hello everybody. Welcome to the Antibody Society webcast series on antibody validation. This is the fourth episode of the series where we're going to look at how we know what antibodies we're working with. The Antibody Society is an international non-profit association. We're an international forum for facilitating discussion, disseminating information, providing guidance, and assisting in the training and advancement of students and postdoctoral fellows. So if you care about antibodies, please join the Society. The speakers in this webcast, experts from academia and industry, are expressing their independent assessments. They're not representatives of the Society. Viewers can write in questions at any time for the speakers to answer, and they will remain online for 15 minutes after the webcast has ended to answer questions. We now know what sort of antibodies there are and what we can buy, but in the second episode of the series, we saw that inappropriately validated antibodies often cause immense, costly and time-consuming confusion, even amongst seriously expert users. This confusion has got several causes. For example, it can be surprisingly hard to discover what antibody you have actually purchased. So, even if you have good web searching tools, such as we heard about in the third episode, and you can discover what the supplier has actually indeed made and characterized the reagents that they're selling to you, and you just have to ring them up and ask them if you're not certain, you may still run into an antibody identity crisis. So in this episode, Anita Bandrowski of the Department of Neurosciences at the University of California, San Diego, the founder of SciCrunch, and Jan Voskuil from Aeonian Biotech are going to explain some of the issues involved in identifying the reagents that you buy. Many companies sell research tool reagents. In the last webcast, we heard about cataloging tools that have identified over 4 million named commercial antibodies. That's a lot of redundancy, not least because some exist in several tagged or labeled forms. But still, there's a lot of antibodies, apparently. In webcast number 3, Jan Voskuil told us about OEMs, which let more than one company market what is theoretically the same reagent, and perhaps they give it a different name. So there seems to be room for confusion amongst you, the users, and that's reflected in the literature. Users often do not unequivocally identify the antibody used in their research. As Andreas Pluktu noted in the first webcast, there are fundamental scientific reasons why this is difficult to do for anything but recombinant antibodies, and this may be one source of the reproducibility crisis that Glenn Begley described in webcast number 2. The inability to reproduce biological research is a drag on you, on the community, and on biological sciences, not to mention the fact that it costs an awful lot of money, and it's usually taxpayers' money that it costs. Journals themselves often do not insist on complete identification of the antibodies that are used, but in fact the issues go even deeper than that, as Anita and Jan will explain today. Anita, you initiated the RRID system to help here. Could you tell us about this and how it can aid the unambiguous identification of the reagents we are working with more clearly?
Sure. Thank you very much, Simon. This is a pleasure for me to be here. So one of the things that I'm going to talk about today, or actually the main thing that I'm going to talk about today, is that RRIDs, these are Research Resource Identifiers, which we truly believe are absolutely essential for rigor and transparency. Rigor and transparency are actually part of new research grant review criteria through the National Institutes of Health. So if you look at this slide, in 2016, the NIH, the National Institutes, has said that there are a few things that are much more important for to look at than novelty. And so they have changed their grant review criteria as of 2016 to reflect the scientific rigor, primarily consideration of things like sex as a biological variable, and of course the authentication of key biological and or chemical resources. And you might ask, why are we talking about this? Well, key resources are things that vary from lab to lab, application to application, and these are the sort of core source of variability in many of these experiments. And so now all grants must include a one-page attachment informing reviewers how they plan to authenticate all of these key biological resources. And of course, what you see there is antibodies are very high on this list of new review criteria, along with cell lines, transgenic organisms, and specialty chemicals. So what this means is NIH really cares about how you authenticate your antibodies, how you identify your antibodies. The problem is that most people, and I would say this is over 50%, were not putting enough information into the paper to even identify which antibody was actually being used. And this is not even at the lot level, this is at the catalog number level. So they would put in something that looked kind of like this. This looks good, and actually this was published just a few days ago on PubMed Central in a very good journal, Nature Immunology. This is a small excerpt of the methods section where it says anti-BRDU 3D4 antibody was purchased from BD Biosciences. And that looks very, very good. It looks like it would identify the product. But in fact, when you look at BD Biosciences, this is a company that has a lot of different products. And it just so happens that this particular 3D4 clone actually has 7 associated products. Now, all of those may actually perform differently. Some may be conjugated, some may be unconjugated. The different conjugates will absolutely affect how this antibody is going to perform in a particular experiment. So when authors don't put enough information into these papers to actually even find the right antibody, it makes the paper not reproducible. So how can we solve this very sort of low-level problem? Well, the way that we can solve this problem is that we have actually had an agreement between initially 25 journal editors-in-chief to try and solve the problem by using the RRID syntax. So the syntax looks like this. The company or provider is being listed as the first portion of how we talk about a particular antibody. The local identifier or the catalog number is another portion of the syntax. The lot number is always suggested to use for the authors. That's the version or the lot that's being used. And then the global unique persistent identifier. So these are persistent identifiers, and I'll show you why a persistent identifier is a little bit better than just asking about the catalog number. The way that this looks in practice is here's an eLife paper, and what I'd like you to look at here is this top of this key resources table. There's an antibody that's identified from the Developmental Studies Hybridoma Bank. This has a catalog number of P1C1 SCAR, and the RRID here is 2618386. And you might say, well, why is that exciting? Well, if you actually click on this particular thing, or if you just look it up in Google, you will find a page that opens up, and you will see a lot of other information that was gathered about this antibody, including the fact that it was used in this particular eLife paper and some others. So this provides additional information to the author, to anybody looking at this. And we know that this works. And the reason we know that it works is because authors have been able to add well over 100,000 RRIDs to their papers since 2014, when the initial pilot project was started. Now, how do publishers know what to do with these RRIDs? So the project has gotten much broader. It has gone much beyond the 25 journals that initially came up with it. But it has also been accepted into the NISO standard for journals called JATS. The version of JATS 1.2, which was released in May of 2019, now contains very specific tags that journal publishers will put in order to actually know what to do with a particular RRID. And this is just the example of the JATS syntax. So We also know from the RRID work that reminders during the publishing process do work well with authors. So if authors are actually reminded, what they do is they do a very good job being able to pull out the information that that is being asked about. So if you look at this bar here, what this says is these lighter shaded bars are the same journal, but just before we started the original pilot project, and this is coming from this bar, that the darker gray bar is coming from the papers after the project has started. And you see this huge shift in the ability for us to be able to identify antibody products. This is again in the same journals that undertook the first pilot, and this was published now some years ago. Okay, so what are the benefits? What are the real benefits of using an RRID over just saying, hey, I can just add a catalog number and a lot number, or as Andrew can put it, hey, I want to only use my recombinant antibody and use the sequence as the identifier. Well, Antibody Registry actually knows which is the source of the RRIDs. It actually knows a lot of things and gathers a lot of data around these antibodies. In this example here, this is an antibody, particular antibody, the NF-kappa B relay antibody, which was originally licensed out of an academic lab by Boehringer. Here's the catalog number. When Boehringer sold this antibody to Roche, along with a lot of its other products, the catalog number was slightly augmented, and this was but you can see the antibody RRID still remains the same. If you look at the Millipore product or EMD product or Chemicon product or now Sigma product, the RRID actually remains the same, but the catalog number has shifted once again, and now this is the Millipore, Sigma, EMD, what have you, catalog number. So a lot of the places where it is very clear that a particular company has sold all of its products to another company, and that other company has changed either the clone ID or it has changed the catalog number, that's fine as long as this ID is there, and then everything in the literature can be associated with this particular identifier as opposed to the individual catalog numbers. But in fact, we know that these are the same. Antibodies are validated by application, and so this is a place where the author would be able to go in and check that in a very consistent framework. Antibody vendors may also pull products off their catalog, off the shelf, if you will, and that is a perfectly fine thing to do. These Boehringer and Roche antibodies are now gone. There's a note a little bit off screen here that says these are discontinued. That's perfectly fine. You can still get the antibody at Millipore. Sometimes you can't get the antibody at all, but at least you know which one was out there at some point that was being used. So there are lots of notes in the antibody registry on independent validation efforts, such as in code. There are lots of RRID publications that are listed, and we tend to have a lot of other notes about how the antibody was created, what kind of standards were used in the manufacturing of it, who was the originating manufacturer, if that is information that is public and known. Now, you can actually find the RRIDs on some of your favorite product catalogs already. Some of the material data sheets also contain this. This is not yet fully integrated into all companies, but we are certainly working towards that. So here's a Thermo page with this Z01 monoclonal antibody, and if you click the little create citation, you can copy this text, which includes, of course, the RRID. Here, if you look at the BioLegend pages, there's the RRID right there. It doesn't suggest at this point this is how you cite this, but it certainly is there as a piece of information. And of course, mouse repositories are also starting to have RRIDs on their pages. Adgene has RRIDs on their pages as well. So this is starting to be a known and well-accepted standard. So as a takeaway, I never thank people, although I am very thankful to all of the people in the lab that have made our work possible. But I would like to thank you, and I also want to call on you to register all of your antibodies. I want to call on you to put RRIDs into your next paper. And of course, I want to call on you to review papers for the methods section. Check those antibodies, see what those authors have done, and you may be surprised at how much or how little that particular scientist had actually done with those methods. Thank you very much.
Thank you very much indeed, Anita. That's super. So the RRID system allows tags to be put on antibody reagents that people have used. If these reagents are monoclonal or better recombinant antibodies, this potentially uniquely identifies them for future researchers. And I think that's a very good step in the right direction. Thank you.
Thank you so much.
The RRID system allows antibody reagents to be uniquely tagged down to the lot used. If these reagents are monoclonal or better recombinant antibodies, this identifies them well for future researchers. However, even if an antibody is carefully identified, you need to be cautious about identifying what it is that you have actually bought. Jan Voskuil will now point out some of the issues involved here. Welcome back, Jan.
Thank you, Simon. This is not just about identification of the right antibody, since we have seen that the same antibody can be featured under different brands, under different ownership, and thus it may receive different RRIDs. And likewise, literature may not be helpful when the catalog number of the successfully used antibody was not mentioned. So, scientists will remain dependent on the information provided by the product datasheet. And therefore, today, I am going to focus on the different contents on the datasheet. When the scientist seeks an antibody from the market, a few demands need to be met. The antibody needs to perform as expected, and when required in the long run, it needs to work consistently from one purchase to the next. And years from now, it needs to remain available for others to reproduce the obtained results. So with this in mind, we need to split the datasheet into two sections and address the different types of information it may provide in each of these sections. So, first we look at the fixed parameters. When ignored, the wrong choice is easily made with profound consequences. All products have a unique catalog number, and when a monoclonal, it should be accompanied with the clone number. The names and symbols need to be consistent with the gene ID and Swiss prot accession. Host species and isotype can be highly relevant, and so are the details of the antigen against the antibody that was generated. Then there is a purification status. There's its formulation, and there's the amount of antibody that you purchase. The second section entails the claims of performance. For example, the antibody is claimed fit for Western blot and immunohistochemistry. There may be a titer in ELISA, specifications of positive controls, and claims of successful use in the literature. There could be data confirming the molecular integrity of the purified antibody. There may be selectivity data. That is data confirming lack of cross-reactivity to other closely related proteins. A real bonus would be the presence of negative controls, but most datasheets show only some positive data obtained in specified applications. So, why are all these specifications so important? It is because of the many pitfalls associated with them. So let's go back to the fixed parameters first to identify the pitfalls. First, certain names or symbols may be shared between two different gene products. For example, the name OCT2 is used for both a solute carrier protein and a transcription factor encoded by two completely different genes. Clone numbers may change as hybridomas are not always stable. Batches and aliquot coding are scarcely used but are essential to identify how a received product may lost its conformity. When you need consistency between purchases, consider buying known quantities instead of known volumes, and make sure you know exactly how the antibody was purified. Both specificity and formulation of the offered product needs to be considered as well. Now let's look at the pitfalls of the performance parameters. Don't just assume that the claimed applications are backed by hard evidence. The scientist has to verify by themselves if the evidence is satisfying. Check also the cited literature to make sure the catalog item mentioned in the materials and methods section was actually used to generate the presented data and not by another antibody to the same protein. Beware that the data need to be consistent with the known cellular compartment and cell types. There is no point buying an antibody showing cytoplasm staining in a breast cancer when it should bind exclusively to a transcription factor in the brain. Make sure the antibody shows fit for the application you need it for. Don't accept data obtained from the right application but with no evidence of specificity, such as, for example, flow cytometry on a single cell line. Beware that experimental conditions within one application can differ between claims. These conditions determine the value of the claim. Ideally, the antibody is compared with a gold standard other antibody. Is compared in expressing and non-expressing cells, and its selectivity is tested by looking at possible cross-reactivity to other closely related proteins, all at the same time and at the same dilution. In summary, it is the responsibility of the scientist to be vigilant and to verify every claim on the datasheet. Only pick an antibody shown fit for the purpose it is needed for. If you need an antibody for flow cytometry, don't choose one showing only a Western blot or immunohistochemistry. Choose the antibody shown fit for the cell types you are interested in and prefer an antibody that shows selectivity in addition to specificity.
Great. Thank you very much indeed, Jan. So as antibodies may have different identifiers in catalogues, they can be, in fact, supposedly identical reagents, multiply repeated. You need to check that they're potentially fit for purpose in the application you propose to use them in. And in particular, you need to make sure that you have adequate positive and negative controls available to help validate the reagents. Jan, I'm sure some of our listeners might be a little bit confused because in the previous talk, Anita told us that the RRID, for example, can follow, is consistent between the same antibody shown between different catalogs. So how can the antibody lose its identity as you described?
Yes, there is a bit of confusion because there are two different mechanisms at which antibodies occur under different brands. One is when a catalog is acquired through an acquisition or a merger into a bigger brand. And then eventually the same antibody changes brand from the original one into the new one. That is something that you can track. And because it's still the same antibody that simply changes brand. but still under the same ownership, as it were. The principle with the OEM is that the ownership changes of the product rather than the brand. So the manufacturer provides their product to different vendors who each are owned by different entities. So the product will end up under different brands, different ownerships, and therefore the product will occur with different RRIDs for that particular reason.
Thank you very much indeed, Jan. So, as you've heard, although antibodies may have different identifiers in catalogues, they can in fact be the same identical reagent multiply repeated. Clearly, identifying the antibody reagents we work with would help reduce uncertainties both for you personally and of course also in the literature. And it seems that the suppliers are not always very helpful here, especially when it comes to revealing batch analysis data. It is critically important that we, the users, unequivocally identify the antibody reagents that we have used to obtain the data sets we describe in our research publications. It's certainly not enough to just state the target antigen and the company from which the antibody came. If we don't do this, for example, using a tool like the RRID system, the reproducibility crisis will destroy us. Once you have managed to correctly identify what you plan to work with, you need to check that it is potentially fit for purpose in the application you propose to use. In particular, you need to make sure that you have adequate positive and negative controls available to help validate the reagents. So in the next webcast, we'll turn to practical issues of physically validating antibody reactivity. And in the first of this part of the series, we'll turn to appropriate use of positive and negative controls with Professor Giovanna Roncador. This is the Antibody webcast series on antibody validation. Thank you very much for joining us, and bye-bye till next time. Dr. Voskuil and Dr. Bandrowski will be online for the next 15 minutes and will be very happy to answer any questions you care to text in. Until next time, from the Antibody Society. If you have a question about what antibody have I found for Anita Bandrowski or Jan Voskuil, simply type it in now at the question and answers tab.